My name is Faulkner. I'm a senior clinical research physician for Synexus. I'm not here to talk to you about specific trials or trials in specific disease areas. My aim here is to give you a little bit more knowledge about trials in general and a little bit more understanding about what different types of trials are out there that you may be interested in the future. Trials can evaluate medical, surgical, or behavioural interventions. They assess the safety and effectiveness of those new interventions, whether they be treatments, devices, changes to diet, but they can also look more at risk factors associated with certain conditions or symptoms as whether they are associated with new conditions. And before a clinical trial starts with a new intervention in humans, years of work will have already taken place on that intervention in laboratories before it comes through to humans. So you will very much not be the first thing having experienced that drug. So there's two main groups of clinical trials, and then we're going to break it down from there. So the one that people most commonly think about are interventional trials. So that is looking at a particular intervention or treatment to see whether it works in the disease group that we're looking at. So whether it's safe and effective against that condition. Then there's observational studies, which aren't so often talked about, and they're aiming to look at the people taking part, but they don't actually influence the treatment they have. They're looking more at risk factors or following people over a course of a condition to watch how that condition progresses over time. And they're not put into treatment groups because there is no treatment with an observational study. So before a full large-scale study takes place, drug companies will often do things called pilot studies and feasibility studies. So pilot studies look to see if a study is even possible. So whether people are willing to take part, whether that be medical professionals or participants, and also roughly how long they think it will take to collect and analyze the information required for that trial to work. And then pilot studies are essentially mini versions of the main study. They test how all the moving parts of the study work together. They test to see whether the investigations that happen before a study starts work in line to give you the correct group of patients to then be able to make the study itself work. And it also can go some way to answering the initial question that was posed by the research and can be included in the results of a main study when those results are published. Now we have different types of trials again. So trials can be used to look at whether a particular treatment can help prevent diseases, and those are called prevention trials. So in that case, we may look at someone who's got a strong family history of a condition such as Alzheimer's disease or cancer, and to see if they can prevent those people from developing that condition in the future. And screening trials look at people who have potentially early signs of diseases before they have symptoms. So they're looking at new methods of investigating and diagnosing conditions in people who are very early on in the disease process and may not even have symptoms at that point, or just very early symptoms and thereby not diagnosed yet. Obviously to see if there's a benefit in picking up the disease early before it has a chance to take hold. Multi-arm trials are very, very complicated. There are lots and lots of different treatment groups within the same study. There will always be one group on the same treatment throughout the study, but it does allow people to change their treatment during the course of the study. So for example, if a new treatment becomes available on the market for a particular condition, a multi-arm study would potentially allow a patient on a trial to look at having that new drug and be still in the trial itself. What that means for the drug companies, they don't have to design an entire new trial every time something else changes within the market, and it allows them to get quicker results rather than having to keep starting from scratch if things are moving within that disease area. It also means that the drug company can stop recruiting people to a particular group. Say, for example, if one arm doesn't appear to be having the same results, they can stop putting new people into that group so they're not unfairly disadvantaged. And it also means that they can potentially take treatments out if it's not working. Observational studies, often called cohort studies, follow a group of people over a prolonged period of time. People who do not have any disease or do not have the disease in particular they're looking for, and they collect information about these people for a number of years. Some of these studies can run for 20, 30 years, looking at people and their risk factors of developing certain conditions. There is no intervention with these studies, as by the title, it's an observational study, but it looks more at risk factors and and genetic disorders of that sort of thing. They are very expensive studies, and these studies are time consuming to run. As I've said, some of them can run for 20, 30 years. But they are used when it wouldn't be possible to test a theory any other way. So if you need to see if, for example, smoking has a link to cancer, we know that now, but how you would do that would be something like a cohort study to see if people who smoke have an increased risk of cancer later on in life. Case control studies are similar. They are, again, observational studies, but they look at groups of people who do have a specific condition and compare them to groups of people who don't have that specific condition. And they look to see whether those groups of people have anything in common, or more importantly, any differences. And that's to try and see if there's a particular thing that the people who have the condition have that the people who don't have the condition don't. The groups have to be balanced. So we try and balance them in terms of age, gender, and other factors that can be balanced for. But it does mean that you can get a look at potential risk factors. However, this is a snapshot in time. It relies more on people's memory, as you're asking people about things that have happened years ago, and that can affect the results. And you also have to be very cautious about whether a cause has happened or whether it's just an association. So the example that's given, people who are on a lower income may have an increased risk of developing a particular condition. It's not the actual money in the bank that causes the issue, but it's more likely to be other things associated. So this is things like more likely to smoke or to have a poor diet associated with it. So there just has to be that caution there. Cross-sectional studies are quick and easy, but the results can be less useful. They will look at people at a set time point to see who has been exposed to a specific risk factor and who has developed a specific disease. So they will look at the people who have developed lung cancer and see how many of them smoked in the past. Again, it doesn't take into account people who are going to develop things in the future, and it also relies on lots of people's memories. So this is often a first step to see if there could be a link, but then they may move on to a more expensive, more detailed trial later on down the road, such as a case control or a cohort study, just to get more data on that point. There's lots of different phases of clinical trials, phase 0 to 4. They are listed on the next slide, so I'll just go to the next slide if that's okay, just so we can talk through each phase individually. So phase 0 trials, the first group of trials of people, and they look at usually a small dose of treatment on a very small group of people, and it's usually only a handful of people, and they're usually healthy participants, so people who do not have the disease that is being trialed with this medication. And they're checking to see if it matches with the laboratory data that's already in process. They're looking at the safety of the data and what happens to the medication within the human body. And these are very small groups looking at very short-term data. They're not looking at effectiveness of drugs at this point. It is a safety issue. Phase 1, we're starting to get a few more people. Again, we're looking more at side effects and safety. And we're also looking more now at getting the correct dosage of medication. So there was often multiple dose arms within a phase 1 study to look at getting the correct levels of drugs within participants to make sure that it's going to be effective. And again, we'll do testing to look at what happens to the different drugs within your body. Phase 2, we're starting to see increased number of participants. So the emphasis is starting to move slightly now and shift. So we're looking more at the effectiveness of the drug, as well as also obviously the safety and the side effect data. The number of participants is often in the low hundreds. And this phase can last several years, with trialing on a few hundred people to start off with, and then they may have to change the dose, or they may have to change investigations to get the correct group of participants, and then they may repeat phase 2 over time to make sure they get the right group of participants and the right drug before they move into the bigger phase 3 studies. But this is starting to look at effectiveness now, and certainly trials do fail at this point because of lack of effectiveness. But safety is obviously also forefront with all of these trials. Phase 3 are the big studies that people become more aware of. So you are looking at potentially thousands of people, looking at different populations, different doses, and also in combination with real life. So how drugs interact with life in general. Is it going to cope if someone has a chest infection, is on antibiotics? Is that going to affect the way the drug works? And it looks more at the long-term effectiveness and the longer-term safety of this medication or intervention. And if the trial is positive and the results prove benefit, then we look at licensing these medications. So the MHRA looks at all of the data that's been found at all phases across the trials, and they will look to see whether they're going to approve the medication or the device or the intervention for use in the general population. Phase 4, this happens once the drug is licensed. Quite often trials will continue looking at much larger populations now that a drug is licensed, and also again over a longer period of time. Some phase 3 trials last 3 to 5 years. These phase 4 trials can go on for 10, 20 years, looking at long-term safety data from these new medications. And it also, again, it's more real world data. This is people who are actually out there, who are taking these medications in their real lives, rather than being more controlled within a trial. And it also looks at rarer side effects. So with more population, you get more likely to find the really nitty-gritty side effects that don't affect very many people. So there's a few steps that clinical trials go through to make sure that the data is as good as possible. And the main three, and that I'm going to talk about today, and this you'll probably be glad to know this towards the end of my presentation, is randomization, placebo trials, and blinding of trials. So randomization. What we're trying to make sure here is that the groups of people receiving the drug versus the placebo, which I'll get to in a second, are balanced. So the details of participants taking part in the trial are put into a computer, and I'm not talking their name, their address, their postcode. I'm talking their age, their gender, their previous medical conditions, any risk factors for the study. And the computer system will then generate identification for them and will balance the groups. So it will make sure that you end up with people of a similar age spread, a similar gender spread, a similar condition spread across the treatment groups within the study. And that can be more than 2 groups. This is just an illustration, but it just gives a more balanced trial. So you don't end up with all of the over 50s in one group and all of the under 50s in another, because that just isn't going to give fair results. And again, I've sort of gone through this already, I'm afraid. So we're trying to avoid bias, essentially, here. We're trying to avoid potentially an investigator thinking, well, this person is more unwell, so I'm going to make sure they go into the treatment group. This person is better, and I don't like them as much, so I'm not going to put them in the treatment group. So it's trying to make the trial fair, and it makes the data more realistic and more fair as well. And the vast majority of phase 3 and phase 2 trials are randomized, and even some phase 1 and phase 0 trials are randomized to a point. Again, to make sure you don't get the same groups, just getting the drug versus a placebo. So there's that word again. So placebo. So a placebo is a dummy treatment. It looks identical to the actual treatment, and it's given in the same way. So if the medication that's being trialed, for example, is a tablet that is green, the placebo will also be a green tablet of the same shape, size, color, so that you can't tell the difference. But a placebo doesn't contain the active ingredient. It is essentially a sugar pill, or, it doesn't have any actual medication within it. And the idea of a placebo is mainly to try and prevent people recording side effects or potential perceived benefits that aren't actually there. So people will potentially, by taking a tablet, develop a side effect purely because they have taken a tablet, rather than it being anything to do with the actual drug itself. So the reason for the placebo group is that if 1 in 10 people on a trial reports having a headache after taking the tablet, it looks to see if it's only 1 in 10 people who are on the treatment phase rather than the placebo phase, or if actually the numbers are similar in the 2 groups and 1 in 10 people in both groups reports a headache, it's less likely that it's going to be related to the medication. It's more likely to be what we call a placebo effect. Now, it's not ethical to give people to a placebo if they really need a treatment for a certain condition. So an ethics committee, which is a group made up of doctors and also lay people, will give permission for placebo to be used within a trial, and they will very carefully look at a protocol, and they will make sure that things are in place to make sure that people aren't disadvantaged unfairly by having a placebo. So you wouldn't get a placebo-based trial for people who have an emergency condition or a significant condition that needs treating right now, because that just wouldn't be ethical. And blinding. So there would be absolutely no point in people having a placebo if they knew they were having one. So blinding is the way that we make sure that neither the participants or actually the researchers know whether a participant is taking the actual investigational product or whether they are taking the placebo. So each participant will be given a code. They are then allocated to for example, a bottle of tablets, and we won't know as investigators, and the participants won't know whether that is a bottle of the drug or whether it's a bottle of a placebo. The idea of keeping me blind is it stops me from biasing as well. So it stops me from saying things like, "Are you feeling better?" to the people that I know are on the drug. It also means that I have to take side effects seriously in everybody, because everybody could be on the drug as part of a trial. Those code numbers are kept secret. However, if there is a medical emergency and I do need to find out whether a participant is on the drug or a placebo, I can do that. So I do have the facility to unblind my participants in the case of an emergency, but I can't just do that because you're nosy. I'm afraid it does have to be an emergency. But at the end of a study, and I've been at the end of a study, which is usually quite some time after you've finished with the study, we do get told whether you've had the drug or the placebo, and you would find that information out later on down the road if you wanted to. And I think that's me done.